Your qualitative analysis, that is where you'll be analyzing salts. Okay, what are the things you're expected to do and to know? I'm going to be giving you some tips, all right, that you can use to answer questions. So maybe you were given certain salts, you do not know what that salt is, but you were asked to analyze the salt. You were asked to dissolve in water. So you are expected to write the test. This test would have been written out for you in the question paper. Add this to water, all right? When you add this to water, filter it in a filter paper. And then what should you do with the residue? What should you do with the filtrate? I'm going to be explaining that to you. Then your observation and your inference. Inference is, what are those things you suspect? Or what are those things that you perceive that is present? What are the ions? And then there is a confirmatory test you will need to do as well to confirm that this particular ion is present. So it could be anions that you're trying to confirm. It could be cations. So I'm going to be giving you a few of these. So the first thing you need to know is when the salt is given to you, all right, they could mix two different salts together. It could be an insoluble salt or a soluble salt mixed together. When you dissolve it in water, so they will ask you, take a portion of this salt, maybe the salt is labeled C or whatever, take a portion of it, put it into water and stir. When you pour it into water, if it's a mixture of soluble and insoluble salts, the soluble one will dissolve in water. The insoluble one will be left undissolved. So you are expected to write, if it is insoluble, you state it there. If it is soluble, you state it whether it's soluble. Now once you have done that, you now take the mixture. They will ask you to divide it. Take a portion of it. Are you listening to me? Pour it through a filter paper so that you can filter. So when you pour through a filter paper, you use your filter paper as a funnel, all right? And then you try to filter. Once you filter, the portion that goes through the filter paper is called the filtrate. The filtrate, that's why you're having filtrate plus. And then the portion that is left on the filter paper that does not go through, that is the residue. Can you see residue? So when you see in your exam, they tell you residue, they tell you filtrate, you should know exactly what they are talking about. The one that goes through is the filtrate. The one that doesn't go through is the residue. So when they tell you add residue to dilute HCl, add filtrate, a portion of the filtrate to sodium hydroxide, to this, to that, that is what you are expected to do. There are some likely questions you'll be seeing in your salt analysis, all right? But I would like to point you to one or two tips that can help you remember or to give you an idea of what the correct ion, maybe it's the anion, the cation, what it should be. Now, when you see green color, you take a substance, a salt, dissolve, you act with water, and then you are seeing green color. Anytime you are seeing green color, if it is green, all right, it is ion 2 that is likely present. Let me give you the key. Can you see green has how many E? Double E. So that is 2. So that's ion 2. But if it's brown color is ion 3. How do you remember? Look at this brown. This W, if you twist it, if you turn it this way, it's going to become 3. So once it becomes 3, that's ion 3. So when you see brown, it's ion 3. When you see green, it suggests ion 2. When you see orange color, it's suggesting chromium is present. Chromium is present in that mixture. Now, when they now ask you the residue, I've told you the difference between the filtrate and the residue. They tell you the residue plus dilute HCl. That means what is left on your filter paper, okay? You scoop it and then you put it in your, you scoop it, all right? And then you put in the test tube. You put in the test tube and then you add a portion of dilute HCl to it. When you add a portion of HCl, when you see blue solution being formed, when you react to dilute HCl, that means copper is present. So blue, copper comes to mind. Now filtrate, that's the portion that has gone into the beaker. The filtrate plus sodium hydroxide in drops and in excess. So in drops, when you see white color, when the filtrate, you have a drop of sodium hydroxide to it and you have white precipitate. There are two things that are suspected to be there. It could be calcium ion or lead 2. Calcium ion, lead 2 ion. Is that okay? When you see white precipitate. Then when the precipitate is insoluble, when that white precipitate is insoluble in excess, that means when you added excess sodium hydroxide, that precipitate did not dissolve. Then it is calcium ion that is confirmed. Calcium ion, you take note, you are adding this base. You are adding sodium hydroxide, which is a strong base. All right, you are adding it to the white precipitate. All right, you are once it dissolves, it is lead 2. If it does not dissolve, insoluble, it's calcium. Let me give you an acronym there quickly that you can use to remember this. The acronym is CANE. What's the meaning of cane? Cane and PBS. C-A-I-N. Cane. What is cane? You still remember, those of you that are Christians, you know Cain and Abel. So cane means when it is insoluble, it is calcium. When it's insoluble, it's calcium. When it's insoluble, it's calcium. So the ions of sorts of calcium are generally insoluble. That is what this thing is trying to show you. So it's insoluble, all right? So when it is insoluble in excess, then excess sodium hydroxide, then calcium ion is confirmed. All right. Now, when you react it with sodium hydroxide in drops and in excess, if white precipitate is found, yes, lead 2, calcium is still suspected. But when the PPT dissolved, when the precipitate completely dissolved, is soluble, then lead 2 is confirmed. Lead 2 is confirmed. So I've told you how you confirm that it's calcium. It is insoluble. That white precipitate is insoluble in excess of sodium hydroxide. It's calcium, but when it is soluble, it is lead 2. And that is PBS. That's the second acronym. So what is PBS? PBS means lead is soluble, while cane means calcium is insoluble in sodium hydroxide solution. Now, when they say this test now, residual plus dilute HCl. So when they ask you to add HCl to the residue, not residual, residue, you are trying to add HCl to test for a particular gas, which is CO2. If you can still recall, one of the properties of an acid is that when an acid reacts, one of the chemical properties of an acid, when an acid reacts with triazocarbonate salt, a gas will be liberated, and that gas is CO2 gas, carbon dioxide. All right. So when you react HCl, an acid, or any other acid, it could be HNO3, you react it with a mixture or with a substance, and then you see some vibration taking place. That vibration is called effervescence. Effervescence occurs. Okay, you will see effervescence taking place. So that effervescence, so when you see the shaking taking place, that's effervescence. That effervescence is because a gas is about to escape from the mixture. So that gas that is escaping is colorless, odorless gas that escapes from that mixture is called carbon dioxide, and that carbon dioxide is from triazocarbonate salt. So when you have the residue plus dilute HCl, colorless, odorless gas, that turns. So this is a confirmatory test for that CO2. CO2 turns lime water milky. What do I mean by lime water? Lime water is calcium hydroxide. So it turns that lime water, it makes it milky. Milky means you see white particles, white powdery particles or patches on the surface of the lime water, and then it confirms that that gas is actually carbon dioxide. So when you see to confirm presence of CO2 gas, you say colorless, odorless gas that turns lime water milky. It turns lime water milky. And blue litmus paper red. Why is it turning blue litmus paper red? Because CO2 is an acidic gas. Are you with me? Gases or oxides of non-metals are acidic. So oxides of carbon, that's carbon dioxide, it is positive to blue litmus paper. It will turn blue litmus paper to red, just the way an acid will turn blue litmus paper to red. But if it is a basic oxide, all right, it's going to be basic. Those are oxides of metals. It will turn red litmus to blue. So, but if it's turning your blue litmus paper to red, that means it's an acidic gas, and CO2 is an acidic gas. So it turns blue litmus paper to red. Therefore, for your inference, CO2 gas from CO3 2-, that's from triazocarbonate 4, is confirmed. Then they can ask you to react with K2CrO4. This is an oxidizing agent, tetraoxochromate. All right. So this is potassium chromate. All right. It's an oxidizing agent, and then it helps to confirm if your D is a reducing agent. So if it decolorizes this, that means this D is a reducing agent. And what are the reducing agents? They are metals. So it confirms the presence of this lead 2 metal. These are another confirmatory test for lead. So when you react D with this K2CrO4, yellow precipitate confirms the presence of lead 2 ion. And this lead 2 ion is a reducing agent because metallic ions are usually reducing agents. Now, you're likely going to test for starch. How do you test for starch? Then maybe the substance is labeled C plus water, and you filter it, partially soluble in water, then colorless filtrate and white residue. When the filtrate, I've differentiated between filtrate and residue. When the filtrate is colorless, all right, and the residue is white. C is a mixture of soluble and insoluble salts, which I've explained. The one that dissolves in water and passes through into the filtrate is the soluble salt. The residue that is left on the filter paper is the insoluble salt. Now, so when they now ask you to react your residue, okay, when you react residue with few drops plus few drops, of iodine solution. Iodine solution. So basically, iodine solution is used to test for presence of starch. It will turn that carbohydrate into blue-black color, or the iodine color will change to blue-black. So a blue-black or dark blue color, it confirms the presence of starch. Starch is present. So with this color change, blue-black, it confirms that what you have here is starch. So these are some of the things that you should have them in mind. You could come across them in your exams, and they will really help you. I wish you all the best in your exams. Bye for now.